everyone, this is Phyo from the FOURCHES lab at North Carolina State University. My research focus is entirely in cheminformatics, where I develop and apply computational methods to solve chemistry problems in the context of drug discovery. A lot of my projects are catered towards the structure class of macrolides and macrolactones, so I want to give a little bit of background on that. So these are some of the azimuth structures of macrolides and macrolactones. They are at least 12-membered ring molecules, and they have a lot of divergent important biological activities. They can be characterized by the presence of an ester functional group within the core rings. You may not have seen these structures before, but you may have heard of their names. For example, this one is Erythromycin. It has antibacterial and anti-inflammatory properties. It is used to treat respiratory tract infections like pneumonia, bronchitis, STDs like gonorrhea, and skin infections. The second structure that I want to show is called ivermectin. It has antiparasitic properties. It may be found as hot guard in the drugstores. It is used to treat heartworms and hookworms in four-legged furry friends. It also used to treat worms and parasites in cattle, cows, horses, etc. Another very important use that I haven't put here is that it's also used to treat river blindness in humans. River blindness causes partial to full blindness with severe skin conditions, and it is used to effectively treat this disease. The last structure that I want to show here is called temsirolimus. It has antifungal, anti-cancer, and immunosuppressant properties. This is used to treat advanced kidney cancer. The reason that I'm showing all these structures is just to emphasize the fact that these macrolides and macrolactones have proven therapeutic properties, and they have a lot of important and interesting biological activities. But what we know about them is still fragmental, and one of the questions that I always get whenever I talk about macrolides and macrolactones is that they look very highly complex, but how do we actually synthesize them? And this is a really good question. Most of these products are derived from nature, so I'm going to show you a process that demonstrates that. So this line is considered an enzymatic assembly line. Each oval is an enzyme, and each colored blocks of enzyme is called a module. These modules produces a certain extender or a certain functional group. So as these molecules get passed from one module to another, those extenders get added, and at the end it forms a very long linear molecule, and it gets cyclized by the T enzyme, forming this macrolide scaffold at the end. In biosynthetic engineering, these enzymatic assembly lines are studied and manipulated and probed to diversify the ending products. It's a really powerful technology. But some of the major obstacles associated with that approach is that there are complications with DNA sequences, and we have limited access to those biosynthetic pathways. The chemical universe of macrolides and macrolactones may be vast. We don't really know much about them because we haven't really explored them. We know for sure, however, that it is highly improbable to synthesize and test all these macrolide analogs to search better, more potent therapeutics. And the ultimate solution to efficiently explore this chemical universe is by applying computational methods. And when we talk about trying to find lead potent therapeutics, we normally apply a computational virtual screening, and it's a very effective technology. So I'm going to show a video to demonstrate that. So in virtual screening, you start out with a virtual chemical library containing 1 million or more molecules, and you run them through virtual screening, and in that process, a lot of these compounds are discarded that may not have the desired chemical properties that we want. And at the end, you end up with a handful of molecules, and they are potential hits, and we study them closely, and we optimize them further so that they have the desired pharmacokinetic properties. The major obstacle of applying this approach to macrolides and macrolactones is that we don't actually have a database of macrolides at this point. So this entire step is entirely missing. This really impedes our exploration and understanding of this structural class. And this is really where my research comes into play, because this is the Williams lab from North Carolina State University. They focus on biosynthetic engineering of macrolides and polyketides. This is my lab, the FOURCHES lab. We focus on the computational aspect of it. So the collaboration between the Williams lab and the FOURCHES lab led to the development of SIME and PKS enumerator. I have already discussed about the PKS enumerator in my second year talk, so I won't be going over that in this presentation. So I'll be mainly talking about SIME. So SIME stands for Synthetic Insight-Based Macrolide Enumerator. So this is the structure of Erythromycin A. It is very well known. It actually launches a lot of other new analogs, macrolide analogs. So this is really useful for treating respiratory tract infections and STDs and also skin infections. So it has a lot of important values, and but it also has some side effects. It has gastrointestinal intolerance and a very short half-life serum. So over the years, a lot of scientists have been working on improving those characteristics, and they found that C8 and C12 are associated with decreased metabolism of this drug, and that these positions C6, 9, 11, and 12 are associated with acid stability. So they have been trying to manipulate the structural fragments in those positions and trying to improve that. And as a result of those multiple years of research, we found Erythromycin. The major difference here between these two structures is at C6. They change the hydroxy group here to a methoxy group here. This is the only modification. But as a result of this compound has a better half-life and reduced gastrointestinal intolerance. Another important finding is the development of azithromycin. So they changed at C9 and they introduced a tertiary amino at this particular position and make it a 15-membered ring. And this is just a minor modification, but in terms of pharmacokinetic properties, it won a long way. It has higher potency, it has reduced side effects, and it also has a much better half-life in comparison to erythromycin as well. And even these compounds nowadays are facing antimicrobial resistance, meaning that they're and as you know, the antimicrobial resistance is happening at an alarming rate. So there is a need to diversify and screen those macrolides so that we can develop novel macrolide and therapeutics in a very efficient manner. Previously, I showed that virtual screening is a really effective computational method to do that, but we still don't have those chemical libraries. We still don't have, especially libraries containing novel macrolide therapeutics. This is still a missing step. But we know what parts of the chemical structures that we want to manipulate. We can leverage that knowledge from different fields. And we decided to develop this SIME software. So we use this knowledge from multiple different fields and try to design novel macrolides based on this knowledge, so that we can find drugs like azithromycin and clarithromycin in a very efficient manner. So in SIME, this is the approach that we're using. So you would have a core macrolide structure, let's say erythromycin A, and then you will identify the position like C10 and 12 that you want to manipulate, and you will provide a list of structural motifs to incorporate at those positions. And you will also identify the positions for sugars that you want to manipulate, and you will provide a list of sugars that you want to insert at that position. So essentially, this is what you will be doing in this software. You have a very good control over the kind of compounds that you want to generate. These are some of the structural motifs that we collected from our first paper. This was published in 2018. That was extracted from 18 experimentally confirmed bioactive macrolides. So currently we're reusing them in our new software, and these are provided in the default. We also extracted the sugars from those known 18 bioactive macrolides as well. And this is also taken from the 2018 study. This is the graphical user interface of SIME, and you can upload a macrolide structures with these positions identified for structural motif replacement or the sugar replacement. And then you can upload a list of structural motifs in those positions. You can also upload a list of sugars as well to be incorporated at the specified positions. And you also have other structural and constitutional constraints so that you can restrain the chemical space further. So you can manipulate these parameters as well. And then you will click generate and it would provide you large chemical libraries of macrolides. So I'm going to show you a case study with the virtual V1b macrolides, also known as V1b, generated with this software called SIME. So these are some of the example structures taken directly from the V1b library. And at a glance, you can see that these are very similar to the already existing drugs. We also did the chemical property distribution. So 1 billion compound is very large. So what we did is we sampled 1 million compounds from those V1b, and then we did the distribution analysis on that 1 million compounds. So we look at molecular weight, mole log P, which is also hydrophobicity, topological polar surface areas, hydrogen bond acceptors, donors, and rotatable bonds. It has a really good distribution, and we could compare it to drug-like groups as well, but at this point we don't feel the need to do that because this is an uncharted territory. But we do have Macrolactone DB at this point, right? Which is also covering a similar chemical scope. So we decided to compare those two distributions. So here, the blue one is from the V1b, the new library that we generated. The yellow one is from Macrolactone DB. The one that I show in the very first part of the presentation. So we compare the two distributions. So the main things that you should look out for is to see if these two graphs overlap, and meaning if they would occupy a similar chemical space. And we found that the V1b is found within the most densely populated regions of Macrolactone DB, which is what we want, which is what we're looking for. So the major takeaway from this slide is that the V1b library showed really well distributed molecular properties that fall within the chemical scope of Macrolactone DB, and this is what we wanted to see. So to conclude this second project, SIME, what it does is that it efficiently enumerates large VS-ready, virtual screening-ready macrolide libraries, and users can specify structural motifs and sugars of choice to integrate at very specific positions in macrolides. This is designed in a way that research findings from different fields can accumulate, and we try to leverage them to generate those very large novel libraries of macrolides to give a very high chance of biosynthetic feasibility. I also wanted to include a quote from our key collaborator, Dr. Williams from the NC State, about the importance and usefulness of this software. So I'm gonna give you a minute to read that. And we also showed that the V1b library had well-distributed molecular property that fall within the chemical scope of Macrolactone DB. And as of now, V1b is the largest in-silico chemical library out there that contains 1 billion fully assembled macrolides with sugars present. And there is a lot more future work that we want to do. We want to computationally model and screen these libraries, and generate new libraries for biological targets that we're interested in, for example, a bacterial ribosome. And this is another project that I wanted to do over the years. I'll probably do that maybe after the defense. So what I wanted to do is I wanted to incorporate gene cluster information for each macrolide. So for example, you will provide a macrolide into this software, and it will use a certain heuristic or an AI-based algorithm, and it will predict the enzyme sequence associated with that. I also wanted to include another feature called synthetic feasibility index in this software as well in the future. So it would be based on a traditional organic synthetic point of view, and also another one based on biosynthetic synthesis point of view. So it could be used as a criteria to restrain the chemical space further and determining what compounds that we're going to be prioritizing.